Hello everybody, welcome to today's webinar. This is Peter Cooper from the NCBI. This is a webinar on human population genetic variation data at NCBI. Lon Fan is going to be our presenter today. And so I'm going to go ahead and turn it over to Lon, and he's going to take you through this information today.
All right, thank you, Peter. Okay, welcome everyone. So these are the topics we'll be covering today. We'll start with dbSNP, and then we'll go into this new project, the Allele Frequency Aggregate or ALFA project, and then we'll do a quick demo on the web and also on the EU to API, and then we'll follow a question and QA. So about dbSNP. So dbSNP just celebrated its 20th year anniversary this year, and currently it houses over 680 million reference SNP from 2 billion submissions. And we annotate the variants on both GRC 3738, and there have been over 500 million RS have allele frequency data. In addition to mapping to GRC 3738, we also annotate the reference SNPs on the mRNA and protein, and also we annotate with functional consequences, clinical significance, allele frequency, and you can find out more about the variant, the RefSNP, other attributes that we annotate and found that link down below. Okay. So the topic today is on the allele frequency. So this is what we currently have in dbSNP. So the name is sort of misnomer in that dbSNP contains both common and rare variants. So this shows you a table of all the projects that currently in dbSNP. And it shows you the project, the number of subjects, and the variant contributed to each one of those projects. So dbSNP aggregate data both from the global survey studies, including NOMAD, TOPMed, EXACT, 1,000 genomes, but we also include regional or cohort-specific studies, including the UK parent and child cohorts, and also the twin cohorts. And also we include regional or national Pacific population. And as we identified more of these studies, we add on to dbSNP. And also including the ALFA project that we include in future dbSNP build, that is in the same scale or one of the largest projects that would be in dbSNP. Okay, so how do you search for this data? You can begin your search by you going to the dbSNP homepage. The search box is on top. There are some examples provided down here. You can search by reference SNP, gene, genome location, clinical significance, and allele frequency range to get you started. Other supported search terms are listed here. If you click on that link, it will take you to the list. Also, there's a handout that you can download. It's the fact sheet that can get you started with the search and displaying the records. Okay. So when you search in dbSNP, when you enter a search term, what you get back to a list of variants in the center here, and then there's some widgets over here to help you manage your filters and search queries. But then on the left here is where all the filters are for all the attributes I described earlier, including the variant types. You can filter by variant types. Clinical significance, and then also, of course, the allele frequency. Okay. So once you get narrow down your search results, on the result, what is shown on the entree display is some summary information about the variant, including its variant type, its map location, the gene, functional consequences, and missense, and also the clinical significance. But then it also included the allele frequency down here. So this is the study-wide allele frequency for each of these projects. But if you want to look at the subpopulation, let's say like from Asian and down genome, you have to go to the reference SNP page, and that you can just follow this link here and go to the full RefSNP page. So the full RefSNP page, again, will provide you again the summary information up here, but the detailed information provided in these tabs here. So there's a tab for each of the categories of information, and you can drill down the information by clicking on the individual tabs. So for frequency, If you go down, if you click on it, you get this big long table that shows you the studies, and then the subpopulation, the studies, and then the samples, and the allele frequency. So if we zoom in on one particular region, for instance here, this shows you down genome studies. It shows you the study-wide, the global frequency data, including sample size, the reference allele and alpha allele frequency, and then also the subpopulations. There are a couple other tools on this this table that kind of help you narrow down to your population of study of interest. That include in a search filter box up here. So for instance, you can just start typing the study name, and then it will narrow the result down to just those that study, and then it shows you the result for that. The other thing we have is the columns here are sortable. So this allows you to sort any of these columns or any of these attributes. For instance, if we sort by the alt or variant allele, either descending or ascending order, whatever your preference is. But in this case, we sorted by descending orders. And what this shows you is that this variant allele is consistently reported or most frequently observed in Asian population across all studies. So that may give you more confidence in terms of prevalence of this allele for your population of interest by looking consistent reporting across different studies. Okay. So you can also download your results by clicking on this here, send to, and there's different formats. The other advantages of using the Entrée system is that you have access to eUtils API, and so all the search that you do on the web, you can do it through the API also and retrieve the results. So we have a GitHub page that you can go to, and there's a demo for eUtils here that you can take a look at, and then put other examples for how to parse and access the dbSNP data. You can also launch that demo on Binder and then just play around with it interactively live. And I'll go over some of the demo later. Okay. So here's an example of the notebook, and essentially there's codes here to access the SNP. Essentially, here's an example. Just enter the same term that you would normally on the web here, and then retrieve the results. The data is also available through FTP download from dbSNP homepage. That's VCF or JSON format. Okay, so that pretty much concludes the dbSNP part of this presentation. So next I want to describe to you about the ALFA project of dbGAP. So dbGAP is a database that's housed at NCBI that contains the results of studies that look at the interaction of human genotype and phenotype. It contains a large amount of data, including over 1.5 million subjects and trillions of genotypes. So dbGAP actually have two control access, open and control. And when it was first started in 2007, the genomic summary result that including p-values and allele frequency was initially open access. However, shortly after it went live, there was a publication that went out that showed that you can identify individuals using a statistical method. You can identify an individual in a study if you have that person's genome. In response, NIH, to protect the participant and also to evaluate the policy, the GSR data was moved under controlled access. However, recently this policy has been reversed. So now we're allowed to once again share the summary result information, including allele frequency. So we embarked on this project called the Allele Frequency Aggregator Project, or ALFA, and this will be aggregating allele frequency in dbGAP and making it available through dbSNP for variant interpretation analysis. So I'll walk you through how we process data. So this will give you an overview of the processing pipeline. So there's all the studies that have been approved for open access, and then it's QC and QA. Then we compute the ancestry of all the subjects in those studies. And then we convert all the data into a standard VCF format. And then they get fed into essentially a dbSNP build pipeline that for normalizing the variant that utilizes a couple of new algorithm and variant representation such as Speedy and Volca. And you can read about that in this publication that just came out this month on those algorithms we use to normalize the variance. And then essentially then once we aggregate the variant and normalize the variant, we can then aggregate the individual genotype and compute the allele frequency, make it available to the normal dbSNP resources. So there's a couple of challenges with this project. One, of course, is dbGaP large volume of data. Then the other one is harmonizing the different subjects and populations across study. Because most of these are self-asserted or self-reported population, or they have no ancestry reported. And the other challenge, of course, is to normalize the variant, because they come in different format, including VCF, Plink formats. They came from different assembly versions. They've been called from different pipeline, and they're of mixed quality. Okay. So to solve one and large volume data and normalize the variant, we utilized the existing dbSNP and dbGaP curations and build pipeline. And to normalize the variance, we used a program called GRAF-pop that is developed by dbGaP. And what it does is it uses the ancestry informative markers to validate the sample ancestry, remove any duplicates, and then also to assign subject with common ancestry to populations. So I'll walk you through briefly how this is done. So this shows you an example of how GRAF-pop assigned the individual to a particular populations. So what GRAF-pop does is it essentially create a genetic fingerprint of all the individual from the genotype and compute the genetic distances for them and plot them on a graph. So what this shows you is comparison between what GRAF-pop computed versus what the self-reported population are in the different colors. Individual with common ancestry are essentially grouped together in a sample shown in these vertices. And then individual with mixed ancestry will fall along the axis. And shown in blue is the 95% interval, where those are the one that with the common ancestry that we assign to a particular population of African, European, or Asian. So here, subject with mixed ancestry, for instance, here there's subject that have been reported as Hispanic, but GRAF-pop actually identified as two separate ancestry. One is that it's a Hispanic group with the Afro-Caribbean ancestry, and one Hispanic group that has more European ancestry. So that's how we assign the individual to different population, and there's 12 different populations that GRAF-pop assign them to. And any individual with mixed ancestry of not within the interval are assigned the others. So we also use the AIM markers to QC the variance and the allele frequency reported by each studies. So we compare the markers from the studies and then compare them to the frequency reported by down genome. So this shows you this is just a plot of the variant allele frequency in the study versus in down genome. Of course, this is a nice they're in a concordance, so that's good. For at least for this European subject. So this shows you the study didn't pass QC because more than half of the variants are reported where the allele had been flipped. So that's why you see this, the axis has been inverted essentially. So it's shown in red here. And then more subtle issues is such that we identified is here where, there seems to be some systematic error where some of these variants where you expect them to be random were assigned at a 0.3 or 0.7. So that failed QC also. And then even more subtle is like where down genome reported the markers, some of the markers as heterozygous, but then the dbGaP study has a marker that reported as homozygous. And typically these are from they derive from microarray assay, and it's probably like that these are probes that failed in the assay, failed to detect the heterozygosity. So that's kind of illustrates some of the process and the step in which we go to the QC, the subjects, and also for the variants. So once we've done all the QC, like I mentioned before, we take all the data, the different format, convert them to the standard input format. And that's just essentially going to, again, dbSNP build pipeline, where then they get normalized and then assigned RS numbers, or if they're novel, or if they assign to existing RS number and then distribute to normal product pipeline. This is a build summary we did recently of some of the about 50 studies from dbGaP that have been approved for open access, and this comprises of over 140,000 subjects and over 500 billion genotypes, and normalized or aggregated to almost 500 million RefSNP. Majority of them are already exist in dbSNP, and then there's about 20 million that are novel. And this is a breakdown of the populations from that build. You see most of it is European, 75% European. There's some representation of the other continental populations. So this is only represent 10% of the potential 1.5 million subject in dbGaP so far, and we'll be adding, increasing. Our goal is to increase the underrepresented populations in the future builds and releases. That may include no cohorts or new populations. And like I said, they'll be included with quarterly with db's new build. And the data for the alpha will be reported on RefSNP page, just like what I've shown you already for the existing data from other projects that already exist in dbSNP. The data will also be accessible by an API. You can get the metadata for the population and the studies. You can also query by position range and also by RS numbers. Of course, the data will also be available by bulk download from the FTP site. Essentially, it's a standard PCF format. We'll have the sequence or chromosome accession, position, RSID, allele, and then there'll be a column for each of the populations with the allele counts. Okay, like I mentioned before, the first release of the alpha data will be coming out in January 2020. So look for that announcement. With that, we'll move on to the demo section of the webinar. So first, I'll demonstrate the Entree search, and then we'll go into the API. So you can start from dbSNP web page, homepage. So I'm going to go ahead and use the example that already is on our help page. So let's go here. So like I mentioned before, there's additional search terms that have been supported. And then there's some examples shown here of some complex query. And we're just going to look at this first example. This is kind of interesting, how you can use a low frequency to interrogate whether a variant is suspect or not. I'll go click on this, and then we'll go ahead and explain in a bit. So typically what I like to do is look at the outliers. So I looked at for variant where the frequency is not detected in any of the projects or as rare. And so here I'm querying actually where the frequency is for 0, right? This suggests that there may be an assembly bias and that there's a, that the assembly may have been incorrectly annotated at that position, so that when they compare it against assembly, we'll call it variant there, but then when they go in genotype, it will fail and they will not detect heterozygosity or low frequency. For instance, if you look at the first one here, on GRC38, the position we reported here, and the allele is CG. And then you look at the allele frequency, everybody sees non-detectable, the variant allele is not detectable, right? But then if you look at the HEVS here, we report both on GRC37, 38. So this is GR38 for this one. You see a C to G here. But if you look at the older version, GRC37, you see the allele been flipped, G to C. So then if you want to interrogate even further, you can go look at the dbSNP web page. You see additional evidence of whether that variant's allele, whether that variant's an artifact or just real. Again, so if you look here, here's GRC37, 38, you see the allele been flipped. But then if you go down to frequency, and then you can also actually see the evidence for this by using the sequence view below by looking at the alignment track between GRC37, 38. So you go to configure tracks, and then if you go to sequence, if you scroll all the way down, we have alignment for all between GRC38 and all the different other alternate assemblies and older version of the GRC assembly. So we go all the way down to GRC37. The alignment between GRC37 38 here, we'll go ahead and add that. There, you can see there's a mismatch that occurred there between tier 7 to 8. Okay. So this is an example of how you can use a allele frequency. That's probably not your normal use case, but it's kind of interesting. So then, okay, now back to dbSNP. We have tutorial how to access the data using eutil on GitHub. So you click on the link to GitHub there. There's a couple of Jupyter Notebook you can look at. There's one that we just recently developed for how to query and parse the allele frequencies called math graph here. And that you can launch this and run them interactively live by clicking on these links. I already had launched one here, so it's right here. Oh, actually, it's, let's see, I'm gonna have to rerun it. So I'll just walk you through briefly what it does. So what this does, so dbSNP is part of the Entree system that allows you to search across not just dbSNP, but other database and link all the information together. So in this example, what it does is search for the gene of interest in gene database, and then it use that information to search the gene to search dbSNP, and then to extract the frequency data. So these are the codes. So here we're searching for searching the gene database first. We're searching for all ACMG genes, and then so what it does is it finds that there are 60 genes. That's here. So this is just some simple widget that we developed just to demonstrate the search. And then from there, it then searches for each of the genes. It search. You can iterate to and search for the variant in dbSNP. So the search for that starts here. It found that there are 832 SNPs in this gene here. This is actually TP57. I'm not sure why the widget didn't show up. Let me try to run it again. I might have to restart it. It meant timeout. Okay, while that's spin up, I can still explain the codes. But anyway, so this allows you to search across multiple databases and integrate the data. So essentially what it does is search dbSNP, it grabs the each of the RRS, then you need to plot, for instance, the one RRS here, the allele frequency. Okay. Is it there yet? No, okay. Oh, it's still running up. Anyway, I guess we can move to the Q&A while this is spinning up, and I can just go back to it later. Do you have any question on that? Okay.
So if anybody has any questions, this is Peter Cooper again. Go ahead and type them in the pod if you have anything to ask right now. We don't have any questions at the moment. Lon, you want to share that other slide, and we can just point out some other URLs that they can go to if they want to. Oh, yeah. So just go to the last slide. Just this last slide has a number of URLs that you can go visit to find out more information. The first two variation services and the speedy notation are useful if you want to understand all the various API things that that Lon was talking about today. Just to remind everybody that we have a blog called NCBI Insights. That's where you can go to find out the latest information about NCBI services and databases, and so we post articles up there several times a week. There's also a Learn page that you can go to find documentation and things like that. We have a very nice set of fact sheets that are PDFs that are on the FTP site. These are the handouts that we give out at conferences, and they'll take you through a particular resource and show you how to do things. And we have a YouTube channel, and that's where this webinar is going to be very soon. And if you want to go there, there's a nice playlist of webinars that you can take a look at, plus some other tutorials. Okay, Lon, we do have one question. Is there a way to check the allele mismatch GRCH38 versus GRCH37 in the dbSNP build 153, the downloaded VCF file?
Yeah, the only way you could do that, I guess, is you compare the annotation on GS3738. We did have a list. If you go to the dbSNP FTP site, we have it in archive, the list of the variant that was that is a mismatch between GS3738. So if you go to FTP download, there's in the pre-build 152, go to human, it doesn't matter, I don't think, yeah, 51, 38. There's known issues. Yeah, so here's the one where it report all the variants where 37, 38 orientations are flipped. So that should not change too much between the last couple builds.
Here's a kind of a general question. Is the rate of discovery of new novel SNPs declining or accelerating?
The number of this declining, I think we'll probably reach somewhere near the threshold right now. Essentially, all the common variants have been found. With all the novel variants, essentially the rare variants, and it really kind of depend on the study size, right? So the larger the study size, the more rare the variant they'll be able to detect. So hard to say, but if I had to guess, it's probably trying to plateau off, like the rate is actually decreasing.
Thanks, Lon. I think you sort of addressed this, but I'll read the question anyway. Can we explore unique variants? Those are individual specific SNPs.
I'm not sure what you mean by unique. You mean like the one specific for populations? Or?
I think he means for an individual, so that would be controlled access data.
It's going to be hard. I mean, you probably, assuming you could, I mean, if you look at the allele count, if it's like 1 or 2, it's probably only coming from 1 or 2 individuals, right? So those are the rare variants. So we could probably try to, let's see, if you go to like, so if you search on dbSNP, if you go something like, we search for really, really rare variants. So we're gonna go ahead and just change this from 0 to, let's say, less than 0.0. So this is 1 out of 10,000, presumably. Let's see if we'll find any. Yeah, it's not many, but there are a few there. So, oh, actually, we should get rid of 0. Let's just go. Try that. Essentially, you just have to search for the really, really, really rare variants. And those will probably, like, if their allele count is like in the 2s or 1s, it's probably because it's only come from 1 or 2 individuals.
And someone asked a question, but really I think they're just making a point that a SNP reported in one individual is probably likely to be an artifact rather than a true variant, which is what you were showing earlier when you were doing the 0 frequency example. Okay, I don't see any more questions. Did you ever get this to run? Okay, let's do that.
Okay, so, okay, it looked like it did run finally. Actually, you can probably run this on other services, like download the notebook and run them on Azure or even Google, which is probably faster. But essentially, yeah, I think that all the codes is there for you to play around with. It was supposed to essentially plot out the low frequency variant, and there's supposed to be a drop-down menu that you can select which variant you want to plot for, and it's supposed to dynamically, you can change the query and then play around with it. But anyway, that's about it.
Yeah, I've got one last one. This will be the last question for today. So they want to find a way to explore SNPs that overlap with multiple regulatory sites, for example, from ENCODE or transcription binding sites. And essentially, you'd have to look at the annotation on the genome to find out where those were, I think. But
very good question. So we do plan on adding the regulatory regions annotations to dbSNP, probably coming out this summer. So then it will be just another attribute that you can just filter over. So it would be just another functional consequence that you see in dbSNP now. So for instance, let's see, let's just go all here, show. So we'll have annotate regular, just like, part of it here. There'll be a list here that will show you all the regulatory regions, including enhancer, silencer, and so on like that. And then we based on RefSeq annotations. RefSeq already have a project to annotate the regulatory regions, so that will be included in dbSNP hopefully later this summer.
Okay, thanks everybody for coming, and thank you, Lon, and I'll go ahead and end the webinar now. Thank you all for coming.